And our final rapid abstract is Sheela Jayaraman from Nottingham is going to talk to us about niclosamide and cyclin inhibitors in a murine model. Thank you, Sheela.
Hello, thank you to the organisers for inviting me to speak today. I'm actually going to talk about a part of the data that was obtained by my student, Grace Martin, as part of her AMMF and University of Nottingham funded studentship. So I don't think I have to explain to this audience that there's an urgent need for new and different therapies for treatment of cholangiocarcinoma. Although there are great targeted therapies out there, of course, the issue about taking targeted therapies is that you actually have to have the genomic alteration in order to avail yourself of those treatments. And so what I've been interested in is trying to find some different treatments for those who can't access those targeted therapies. So I came at this with the idea that, of course, cholangiocarcinoma cells are proliferating very fast, so very likely their cell cycle is going to be dysregulated. And I myself and others have looked in the TCGA datasets and seen that about 40% of cholangiocarcinomas have genomic alterations in either cyclin D or CDK4 or 6, or indeed the inhibitor for CD4K and 6p16. But more interestingly than whether or not you have the genomic alteration is the fact that those genes are upregulated in tumours compared to non-tumours. So we know that we're expecting that cholangiocarcinoma cells are going to have dysregulated cell cycle. And just to remind everybody, of course, I'm sure most of you are very much aware, but I'll just go through very quickly. High CDK4/6 activity will, of course, result in high levels of phospho-Rb, and this allows release of E2F and then initiation of the first step in the cell cycle. So you get faster G1S and increased proliferation. With this in mind, we thought we would try some of the CDK4/6 inhibitors to see whether cholangiocarcinoma cells were sensitive. And we chose palbociclib because it's already used in the clinic to treat hormone receptor positive HER2- metastatic breast cancer. So we thought this inhibitor should be easily translatable. So we looked at a variety of different tumor cell lines, and they have different genetic backgrounds, they have different etiologies, and we looked to see whether they would be sensitive to palbociclib. And in fact, the three that we looked at were all sensitive to palbociclib. But perhaps even more interestingly, it was the fact that normal bile duct cells are not sensitive to palbociclib. So that's really rather important. Okay, so we thought, great, palbociclib is used in the clinic, maybe we can translate it. However, something that we started to think about was the fact that often patients are sensitive to palbociclib initially, but then there is quite a variety of mechanisms whereby you can develop resistance. And I'm not going to go through all these different mechanisms today, but we started to think that perhaps combination therapy would be a better way forward because that is very much considered the best way to treat tumours nowadays. And in addition, we had also come across a study which had shown that just in a very tiny little study with advanced pancreatic or biliary cancer, palbociclib monotherapy hadn't been hugely successful. So with that in mind, we thought, well, okay, what other drugs might be useful to block cholangiocarcinoma cell viability? And we decided that we would carry out a screen with a drug repurposing library with about 100 off-patent drugs. And of course, drug repurposing libraries are useful because those drugs are already known to be tolerable in man, and they've been used to treat other conditions. And also, if they're off patent, they're generally quite cheap. So, the drug that was our major hit was niclosamide. We did get 5 or 6 good hits, but niclosamide was the most useful. And we went and looked to see whether these different tumours, cell lines would be sensitive to niclosamide. And again, they showed reasonable sensitivity to niclosamide. And again, the bile duct epithelial cells tended to be less sensitive to this drug. So what is niclosamide? Well, it's a well-characterized anti-tapeworm medication, and it was identified in the late 60s, and it's been used worldwide since then for many decades. So again, it should be reasonably translatable into the clinic, we thought. It's known to depolarize the mitochondrial membrane, so decouple electron transport from ATP synthesis, so the cells are not producing ATP. And it can also release cytochrome C to the cytosol to bring about apoptosis. And importantly, it can inhibit multiple signaling pathways, and I think that's very key. It's not reliant upon just hitting one specific target. And it can, at least in some tumor cell lines, it can increase protein ubiquitination. So bearing all of that in mind, we went and had a look to see whether in our tumor cells what is happening. And we stained our tumor cell lines with mitotracker red, which stains polarized mitochondrial membranes. And I think you can see that there is less red stain with more niclosamide concentration. So yes, we are getting a decrease in membrane polarization in our tumor cells, and to some extent also in the bile duct epithelial cells. We then went on to have a look at protein ubiquitination, and we could see when we use an antibody against a pan-ubiquitin antibody in Western blotting that when each of the cell lines was treated with niclosamide, you could see an increase in the ubiquitin track in each of these cell lines, and you can see this shown here in these bar graphs. And interestingly, there's a sort of trend. The cells that were more sensitive to niclosamide seem to also have the greatest ubiquitination. So what does ubiquitin do? Well, it is a small protein that is added to other proteins, and then when they become modified, it targets those proteins for the proteasome. So that leads to protein degradation. So we wondered whether any of the proteins that we might be interested in that are involved in the cell cycle might be downregulated. And what we saw was that cyclin D1, which is the partner for CDK4/6, was very strongly decreased in the presence of, niclosamide, as is beta-catenin. And in fact, our favorite transcription factor in my lab, which is an oncoprotein in cholangiocarcinoma and also promotes the cell cycle, is also degraded by niclosamide. But it's not just causing degradation of everything. It does allow there to be cleaved caspase 3, which is increased in the presence of niclosamide. Okay, so all of that's very interesting, but the question is, do the two drugs work together? Does niclosamide and palbociclib have a combination effect? And to answer that question, we carried out combination index experiments where we put the two drugs in separately or together and looked at cell viability. And you can see at different doses, the combination, which is in blue, has a greater effect than each drug alone. And just to explain, these experiments tell us that we have a synergistic effect by putting these two drugs together rather than just an additive effect that you might expect if you put two drugs in at the same time. And you can sort of see this in what's happening with phospho-Rb. You can see that palbociclib is reducing phospho-Rb, as you would expect, since it's blocking the kinase that causes the phosphorylation. Niclosamide, interestingly, is also reducing phospho-Rb, and the two drugs together are very strongly decreasing phospho-Rb. So just to say, this effect occurs in other cell lines. It also, we looked to see what is happening to cell cycle and also to apoptosis. And we saw that each drug alone increased G1 arrest, but the combination is better. And each drug alone had small effects on apoptosis, but the combination is better. So we had a look in 3D spheroid models, and again, the combination is better than each drug alone. And perhaps just as interestingly, the combination and each drug alone don't have very much effect on normal bile duct epithelial cells grown in this similar sort of spheroid type of model. Okay, I'll get to the end. So the important thing is, does it have any effect in a xenograft model? And we were able to show that when we grew the tumors to 3 millimeters and then treated them with either niclosamide alone or palbociclib alone, both drugs have considerable effects on tumor growth, but the combination was the most effective. And you can see this most clearly here. It's the combination that has the most significant effect. And you can see this also in tumor size. Here, this is the combination compared to the others. So, and this was found to be indeed a synergistic effect. So to finish, just to say, I don't need to go through all this, these two drugs seem to work well independently. They also work well together, and they have a greater effect when they're put together. They are used in the clinic for other ailments, and I'm hoping that they should be easily or more easily translatable. And I think it would be interesting to see if we could translate that to the clinic. And I'd just like to thank Ka Ying, who was another student in the lab who helped with this work, as well as particularly Grace, Martin, and Gagan, who was a master's student, and Sebastian, Kevin, and Farhat, who I collaborated with. Thank you very much.